Host defenses consist of two categories: the innate or non-specific defenses and the adaptive or specific defenses. The human body has many innate defenses to prevent disease. These include the first line defenses such as the skin and mucous membranes, phagocytosis of microorganisms, inflammation, and the complement system. The human body has two main divisions of adaptive defense: humoral immunity and cell mediated immunity. Antigen presenting cells aid in the activation of these defenses. A humoral immune response consists of the activity of B cells and the antibodies they produce. Cell mediated immunity is a host defense used to fight intracellular pathogens and abnormal body cells, such as cancer cells. The protagonists of cell-mediated immunity are T lymphocytes, which can be divided into two groups: helper T cells and cytotoxic T cells. Helper T cells secrete cytokines to activate other cells of the immune system, while cytotoxic T cells attack infected cells directly. The innate and adaptive components of the immune system work together to carry out host defenses. Host defenses can be divided into two categories: innate or non-specific defenses and adaptive or specific defenses. These defenses act together to protect the host against infection. The human body has many innate or non-specific defenses to prevent disease. These include the first line defenses such as the skin and mucous membranes, plus the second line defenses such as phagocytosis, inflammation, and the complement system. All of these defenses act against all pathogens and are not specific to a given pathogen. The third line of defense is comprised of the adaptive or specific defenses. These defenses recognize a specific pathogen and must be activated by antigen presenting cells before they can work. These defenses include antibodies and cytotoxic cells. Together, these defenses make it very difficult for pathogens to penetrate, colonize, and cause disease. Chemotaxis is the movement of a cell toward or away from a chemical stimulus. Phagocytes use pseudopods to move toward microorganisms or damaged cells at the site of infection, often arriving only minutes after infection. Chemotactic chemicals that attract phagocytes include microbial products, components of damaged cells, chemicals released by other white blood cells, and peptides derived from the complement system. The plasma membrane of phagocytes usually attaches to glycoproteins on the microorganism's surface. This process is called adherence. Adherence is made easier through opsonization, a process in which antibodies or complement proteins from the host coat the microbe's surface, serving as handles so that the phagocyte can more easily attach to the microorganism. The proteins that coat the microbe are called opsonins. The pseudopods fuse, forming a sac called a phagosome. The phagosome enters the cytoplasm, where it fuses with lysosomes that contain digestive enzymes and other antimicrobial substances. The fused structure is called a phagolysosome. Digestion of most bacteria is complete within 10 to 30 minutes. After phagocytosis, the remaining debris is eliminated from the cell by exocytosis, in which the phagolysosome fuses with the plasma membrane and expels its contents. Some of the contents of the phagolysosome may also be presented on the cell membrane through a process called antigen processing and presentation. Let's review the steps in phagocytosis. The pseudopods of a phagocyte use chemotaxis to approach the microorganism. The pseudopods adhere to opsonins that are attached to the phagocyte. The microbe is ingested within a phagosome. The phagosome fuses with a lysosome to form a phagolysosome, in which digestive enzymes break apart the microorganism. Finally, the remaining debris is eliminated by exocytosis. Parts of the microorganism are presented on the surface of the phagosome. The human body has many surveillance mechanisms for detecting pathogens. Phagocytes are able to detect foreign cells by looking for molecules on the surface of those invaders that are not found in the human body. Adaptive defenses, such as B cells, antibodies, and cytotoxic T cells, are looking for a specific antigen on the surface of the invaders. If a microbe can hide from these surveillance methods, then it's more likely to reproduce within the body. When immune surveillance cells, such as macrophages, are looking for invaders, they search for surface molecules that are not found on host surfaces, such as peptidoglycan. Capsules that surround some bacteria are usually composed of polysaccharides similar to molecules found on host cells. Thus, when a macrophage encounters an encapsulated bacterium, it doesn't recognize it as foreign and ignores it. This is a strategy used by Streptococcus pneumoniae, a common cause of bacterial pneumonia. Even if a bacterium is engulfed by a phagocyte, it may not necessarily be killed. The tuberculosis bacterium is able to survive within a phagocyte by preventing the fusion of lysosome and phagosome. This keeps the digestive enzymes away from the bacterium. Bacteria such as Shigella and Listeria can actually escape the phagosome and live within the cytoplasm of the phagocyte. Even if the phagosome does fuse with the lysosome, some microbes are not affected by lysosomal activity. Leishmania, a protozoan, is able to resist the lysosomal enzymes. Legionella and some staphylococci inhibit the pathway that leads to oxidative killing within a phagocyte. Some bacteria, including the tuberculosis bacterium, actually reproduce within the phagocyte. Several different types of microorganisms are capable of hiding from host defenses by changing their surface antigens frequently. These changes may occur as a result of mutations or genetic recombination. RNA viruses such as influenza virus or HIV frequently exhibit antigenic variation on their surfaces. Trypanosoma, the organism that causes sleeping sickness, has a surface glycoprotein that undergoes frequent changes. In all of these cases, it is the survival of the fittest. The microbes with the undetectable antigens are the ones that are least likely to be detected and most capable of reproduction. Successful pathogens have evolved methods for inactivating the host defenses that would normally lead to the pathogen's destruction. This gives the pathogen a better chance of colonizing and reproducing within the host. Complement is the group of proteins that, when activated, undergoes a cascade of reactions leading to inflammation, opsonization, and cell lysis. Some bacteria simply evade complement by covering the complement binding sites with a capsule. Other bacteria have peptidases that destroy key complement proteins and block the cascade. Neisseria and some viruses mimic a control molecule that the human body uses to keep complement inactive when it's not needed. Many bacterial pathogens, including Pseudomonas and group A streptococci, produce molecules called leukocidins. Leukocidins cause destruction of phagocytes, such as neutrophils and macrophages. Species of Neisseria that cause meningitis and gonorrhea secrete an enzyme called IgA protease. This enzyme destroys a class of antibodies called IgA that are located in mucous membranes. As a result, these bacteria are much less likely to be detected in these regions. Another way in which a pathogen can inactivate host defenses is more general. Several viruses, most notably HIV and the measles virus, are able to cause generalized suppression of the immune system. Often, these viruses work by suppressing the activity of cytokines or by reproducing within macrophages and T cells. Some bacteria produce toxins that are superantigens. These toxins distract the adaptive defenses, causing them to produce a nonspecific exaggerated response and fail to respond to the actual invader.
Damage to the body's tissues through heat, chemicals, sunburn, cuts, or microbial infections triggers a general, nonspecific defensive response called inflammation. Signs and symptoms of inflammation include redness, pain, heat, swelling, and sometimes loss of function. Inflammation has three main functions: to destroy the agent causing injury, to limit the effects of the agent on the rest of the body, and to repair or replace damaged tissue.
The complement system is a set of proteins that circulate in the blood serum. When activated, these proteins work together to destroy foreign cells by cytolysis, to activate the inflammatory response, and to assist in phagocytosis. The main components of the complement system are called C1 through C9. Complement can be activated through three different pathways: the classical pathway, the alternative pathway, and the lectin pathway. The classical pathway was the first activation pathway of the complement system to be discovered. The classical pathway begins when a pair of antibodies attach to antigens, such as proteins or a large polysaccharide on the surface of a bacterium. The antibodies bind and activate C1. C1 then cleaves several copies of C2 and C4. Together, the C2A and C4B fragments form an enzyme that cleaves C3, activating the complement system. The alternative pathway of complement activation was so named because it was discovered after the classical pathway. While the classical pathway requires antibodies specific to the invading microorganism, the alternative pathway requires three complement proteins called factor B, factor D, and factor P, or properdin. These proteins are attracted to microbial cell surface material, such as the lipid carbohydrate complexes of certain bacteria and fungi. Together with C3, these proteins result in the cleavage and activation of C3 proteins, which in turn activate the rest of the complement system. The most recently discovered pathway of complement activation is the lectin pathway. When macrophages ingest material through phagocytosis, they release chemicals that stimulate the production of carbohydrate binding proteins called lectins. The lectins bind to distinctive patterns of carbohydrates on the surfaces of certain bacteria and viruses. The lectins themselves can act as opsonins for phagocytosis, but also activate C2 and C4, which leads to the activation of C3, leading to the activation of the rest of the complement pathway. Once the complement cascade is activated, several events occur that ultimately result in the destruction of the pathogen. C3A and C5A act as chemotactic factors, attracting phagocytes to the site of infection. C3A and C5A also bind to mast cells, triggering the release of histamine and other chemicals as part of inflammation. C3b acts as an opsonin, coating the invading cell. This coat makes it easier for the phagocyte to adhere to the invading cell during phagocytosis. C5b binds to C6 and C7, which attach to the plasma membrane of the invading cell. C8 and several C9 molecules bind to the C5b, C6, C7 complex, forming a cylinder shaped membrane attack complex, or MAC. The MACs form circular holes in the membrane of the invading pathogen, causing water to enter the cell and ions to leave. This results in cytolysis, killing the cell.